Hi, and welcome to Psychology 2. We're now going to look at development. Now, psychologists study development in order to understand the changes that humans experience from conception to the end of life. Now, because development covers such a long time span, developmental psychologists typically specialize by limiting their investigation to a particular period of life, such as infancy, childhood, adolescence, or adulthood. So within any of these age stages, psychologists may focus on different aspects of development: physical, mental, social, or personality development. Now remember from chapter 1 that psychology seeks to describe, predict, explain, and control behavior. Those are the 4 goals of psychology. If psychologists can accomplish these goals, then we can promote health development and prevent, or at least the maladaptive patterns of development. We can prevent many of them, or at least do some correction. Developmental psychologists seek to learn the causes of developmental abnormalities. For example, should a pregnant woman sustain from smoking and drinking? Well, yes, they should, because of the effects that it can have on the developing fetus. Some developmental psychologists focus on adult development. For example, what factors are important for maintaining a healthy relationship? What conflicts and/or changes can we expect as we develop through our 30s, our 40s, and our 50s and onward? What about the development as an older adult? The information acquired by developmental psychologists can help us understand how our physical, social, and cognitive development unfolds and explains factors that are required for more normal growth. Perhaps knowing how we develop can also help us make decisions about how we rear our children and lead our own lives more fully and completely. So let's look at human development. Now in part 1, we're going to include looking at more closely at what is developmental psychology. Hereditary, we're going to look at prenatal development and physical development and learning. Now in part 2, we're going to cover the cognitive stages of development, socialization, and social relationships with the special concerns towards later adulthood. So let's begin. When we first looking at developmental psychology, they like to identify 3 key controversies, if you will, or controversial issues in the developmental psychology field. One is, to what degree do heredity and environment influence our development? 2, is development continuous, or is it in stages? And then 3, to what extent are personal characteristics stable over time? You know, who you are as a teen or as a child, does that stay consistent through time? Or do we, as we age, change some of our own personal characteristics over time? Let's start with number 1. To what degree do heredity and environment influence development? Now, some developmentalists, they take the view that our abilities are determined almost exclusively by our heredity and transmitted through our genes is one of the main things, while others maintain it's the environment that determines what or who we become. Now this has been known as the nature-nurture controversy. Which came first, or is it a combination of the 2? Secondly, is development continuous, or does it occur in stages? Well, developmentalists ask whether aspects of development are best understood in terms of gradual, continuous, cumulative changes, or in spurts in the form of stages, with one stage quantitatively different from the next stage. And this is all part of the sort of ongoing research in developmental psychology. Now, the third point that we touched on, as the third question, is to what extent are personal characteristics stable over time? And again, here, developmentalists also ask whether certain personal traits, such as intelligence, aggression, or aspects of temperament, tend to be stable, or are they changeable over time? So, If we're exploring further to investigate age-related changes, and developmental psychologists tend to use a longitudinal study. Now, a longitudinal study is one in which the same group of participants is followed and measured at different ages. So, a longitudinal study of children in age 5, a group, let's say you have a group of 50 kids age 5, you'll check in on them at age 10, age 15, age 20, age 25, age 30. That's a longitudinal study. Now, the other means by which a lot of developmental psychologists will work with what is known as cross-sectional studies. Now, in cross-sectional studies, it's which researchers compare groups of participants of different ages with respect to various characteristics to determine age-related differences. So instead of looking at over a long period of time of one group, they'll look at a variety of people in the same issue. That's what is known as a multicultural perspective. Now, the benefits of the longitudinal study include being able to map the stability of variability of all kinds of characteristics throughout the developmental phases. So if we were trying to track personality traits at a 5-year-old and how might they change or be stable over time into middle age and a late adulthood, then that longitudinal study would help us an awful lot. Now, the drawbacks, well, that includes that it's time consuming. Of course, the researchers would have to be keeping track of data over that lifetime of those individuals or passing it on to other people. So it's time consuming and it's expensive. People may drop out of the study over time, leaving the researchers with a biased sample. If people, as they get older, they die at different ages, or they opt out because it's no longer something they want to participate in. Now, one of the benefits of cross-sectional studies are that they are less expensive and less time-consuming. It's a method for researchers to compare groups of different ages at one point of time. Now, the drawback is that it cannot determine outcomes over time. Now, there may be issues of cohort differences which do not come up in the longitudinal study. Now, one of our development as individuals begins before we're born. And we'll start at the beginning here in this section, and more specifically, we're going to begin with heredity. Now, hereditary traits are transmitted through our genes, which are located on each of our 23 pairs of chromosomes. Now, at conception, the sperm adds its 23 single chromosomes to the 23 that are with the egg. Together, they form what is known as a zygote, and it contains the full 26 chromosomes, or 23 pairs now, containing all the gene material needed to create one of us, a human being. Now, 22 pairs are known as autosomes. The remaining pair are called sex chromosomes, and they carry the genes to determine our sex. Now, primarily, that's primary and secondary sexual characteristics and the sex-linked traits, and you'll see those in your textbook. Now, sex chromosomes in females have 2 X chromosomes, and males have an X and Y chromosome. Within the gene, there are dominant and recessive genes that influence traits that we'll see materialize in us. Now, in my day, when I was taking university, that was a course that I took on genetics, and most notably, following traits through genetic sequencing, and in particular, looking at the role of dominant genes and recessive genes. So let's look at those. Dominant genes are genes that are expressed in the individual. So if you have brown hair, if you have there's a variety of traits that you will have as a result of you having a dominant gene for that particular trait. Now, the dominant genes, they control the expression of the trait. So if you have a dominant and a recessive gene for hair color, it's the dominant gene that will express itself. There is another way for a gene to be expressed, and we'll get into that. Now, for example, an individual having one gene for brown hair and one gene for blonde hair will have brown hair if the gene for brown is dominant. Now, we'll come back to this in a minute. Recessive genes, and these genes will not be expressed if paired with a dominant gene. So the dominant gene always wins out. You can have 2 dominant genes or a dominant gene and a recessive gene. In this case, it's the dominant gene that gets expressed. Two dominants, of course they will be expressed because they're the same. But if you have a dominant and a recessive gene, that dominant gene will do. But if you have two recessive genes, then the recessive gene comes out. Now, as in the case of hair color, brown hair is generally more dominant, recessive is blonde. But if you have two recessive genes, you will have blonde hair. So these genes will not be expressed when paired with a dominant one, but will be expressed if paired with another recessive gene. Now, traits of recessive genes are only expressed when paired with other recessive genes of the same trait. And that's an important note. So two recessive genes express the trait of blonde hair will result in a person having blonde hair. Now, there are 3 stages in prenatal development. Now, we're moving from the sort of genetic component to saying, okay, now that we know that these are the building blocks, then let's go into the prenatal development to see how these building blocks materialize. And there are 3 periods that we're going to look at, or 3 stages. Now, in the image that I have, it'll include the 4th stage, or the first stage really, which is conception. And then the next stage, or what we'll see in our textbook, is the period of zygote. About 2 weeks, so from at conception, the zygote travels through the uterus or to the uterus and attaches itself to the uterine wall. In the first 5 to 6 days, the zygote engages in cell division and produces 2 parts, the fetus and the placenta. Zygote size is about the size of a period in your textbook. Very tiny, much work is being done, but that's the first stage after the insemination, and the egg and the sperm cells have put together into one, and then the zygote is what you've actually got, and it starts in this early stage with cell division. Now, the second stage, or the period of embryo, week 3 to about week 8, major systems, organs, structures of the body develop. Size at the end of this stage is about 2.5 centimeters in length and weighs about 4 grams. So in the first 8 weeks of conception, we've gone from something the size of a period on the page of your textbook to something 2.5 centimeters that has the semblance of and the beginnings of major systems, organs, and structures of the body. And then the third period, or 4 back here, excuse me, is the period of fetus. Now, 9 weeks to about birth, the bone cells begin to form, and it's time for rapid growth and further development of the structures, organs, and systems of the body. Now, there are examples, and you might be aware of people who fit within this, where multiple birth, that is, more than one embryo, is produced at the same time. Now, most multiple births are twins. And there are the identical or monozygotic twins, where there's one egg been fertilized by one sperm. However, the zygote splits and develops 2 identical embryos with identical gene material. Now, fraternal or dizygotic twins, instead, they develop 2 separate eggs, which are related from two eggs that are related from the ovum, or relayed from the ovum, fertilized by 2 separate sperms. And in this case, the twins are as genetically different as any other 2 siblings might be from the same family. They're no different than brothers and sisters that are 2 years apart. Essentially, you have 2 individual people with their own genetic material that they don't share any more than what siblings do. So, interesting. So there are some potential negative influences to prenatal development, and these are known as teratogens. So when we look at teratogens, these are harmful agents in the prenatal environment that can have a negative impact on prenatal development and even cause birth defects. Now, the damage caused depends on the intensity and when this teratogen is present during the prenatal development. Now, what I'm going to do is just going to review some. You'll find them on the chart behind me now. Most teratogens, drugs, disease, environmental hazards such as X-rays or toxic waste, do the most harm during the first trimester. Now, a trimester is the first 3 months. If you think of a 9-month birthing period. The first trimester is the 3 months, the next 3 months is the second trimester, and then the third trimester. So most of these teratogens are most harmful, if you will, in the first trimester. Now, teratogens such as viruses like rubella can cause hearing, sight, and even intellectual disabilities, heart defects, and damage to central nervous system if the mother contracts it in the first trimester. As mentioned just a bit ago, of course, the first trimester, a lot of things can make or break the success of a healthy born baby. Prenatal malnourishment can also negatively affect development and have particularly harmful effects on brain development during the final trimester. The use of heroin, cocaine, crack has been linked to miscarriages, prematurity, low birth weight, physical defects, and fetal death. Women who drink alcohol heavily during pregnancy risk having babies with fetal alcohol spectrum disorder, FASD. Now, babies with more severe cases are intellectually disabled and have abnormally small heads with wide eyes, a short nose, and other anatomical abnormalities. In addition, they exhibit behavior abnormalities such as hyperactivity. And so the effect of that excessive use of alcohol during pregnancy doesn't just affect the child's sort of cognitive and intellectual components, but it also deals with and affects their physiology and what affects, how that affects the overall development of a fetus. It's pretty significant. Smoking has been associated with higher rates of mature birth, spontaneous abortion, stillbirth, infant mortality, and sudden death syndrome. Caffeine has been reported to increase risk of miscarriages. So how will we now, so now we're going to move into the area of physical development and learning, and we're going to explore from infancy to adulthood. The first group, if you will, that we're going to look at are neonates, the newborns. That a neonate is a term used for newborns up to about 1 month old. They have a pretty good range of reflexes in order to navigate the world around them. Now, reflexes are built-in responses such as sucking, swallowing, coughing, blinking, rooting, and it's the first in this set of important behaviors that they need to be successful. Now, they're present from birth, and some disappear between the 2nd and 4th month as the brain develops. Now, 5 senses, although not fully developed in an infant, are functional at birth, and the newborn already has preferences for certain odors, tastes, sounds, and visual configurations. Now, hearing is much better developed than vision is in the early days. Infants can turn their head in the direction of a sound and show a general preference for female voices, especially their mother's voice. They favor sweet tastes and are able to differentiate between salty, bitter, and sour solutions. And they're sensitive to pain and responsive to positive touching, hugging and cuddling. Now, vision, on the other hand, it's at about 20 to 600. Now, we're used to having, or at least in our adult life, when you have very good vision, 20/20 vision, but 2600 is very poor. And then about 20/20 is reached at about 2 years old. Now, newborns focus best when the object is about 9 inches away and prefer color stimuli to gray. Now, although they can't distinguish all colors. Now, visual cliffs. This is a research piece that has been done for many years, and it's an apparatus used to test depth perception in infants and young animals. Most babies ages 6 to 14 months can be coaxed to crawl across a shallow side. Now, you have to sort of imagine this is a room where you're got a raised part here and it drops off significantly, but there's glass across the top, and so a child can go across and not fall. But the test is to say, when they are coaxed across, will they go? Well, apparently they'll move between, when they're 6 to 14 months, they'll be coaxed across a shallow side. So, a glass that's, say, this high off the ground, they can be coaxed across. All right? Babies prior to 6 months will cross onto the deep side. The depth perception isn't there, and certainly as they get older, the recognition of depth becomes stronger. Now, researchers concluded that visual discrimination begins with infants crawling, so that that component of movement, where movement and visual cues, that you need to stop the movement when the visual cues indicate a drop. So that relationship between needing those skills and crawling are correlated. So what about learning in infancy? We've looked at the sort of physical parts, but let's look at learning. And when we present with a new or interesting stimulus, infants respond by generally becoming quieter. Now, what's happening? Well, their heart rate starts to slow. They start to fixate on the stimulus, whatever's being presented in front of them. But when they become accustomed to this stimulus, they stop responding. This is what is called habituating. If that stimulus is paired with a new one, the infant will usually pay more attention to the new one. Now, this indicates they remember the original stimulus but prefer the new one. Babies only 42 minutes old can imitate gestures such as sticking out their tongue. Now, that's an important component when you look at the overall perspective of how might infants learn things and learn familiarity. It might be through this imitation. So let's look at physical and motor development. As you may be able to guess, that most obvious period of growth, physical and motor in particular, is during infancy to adolescence. That period has the most growth, development, and cognitive, physical and motor growth, as well as some cognitive. Now, it's an important distinction here. Some changes are to do with learning. However, there are some which are not to do with learning, but are to do with maturation. Maturation is naturally occurring changes as a result of our own genetically determined biological timetables. There's no studying required. It's what's going to happen, like walking, and how strongly upright you sit as an infant. Those are things that are maturation. They are things that will unfold naturally when those times come and as they start to be, we get stronger. So in infancy, many motor milestones such as sitting, standing, and walking are primarily a result of maturation. Interestingly, the sort of sequences by which basic motor skills emerge is this way. So we move basically from our head downward through the trunk and legs. Therefore, lifting our head before sitting and walking is why that happens in the stages that it does. And that's maturation. As your muscles gain some tone and you gain control, the first ones are head, then trunk, then legs, which is why we begin with lifting our head, then we get into sitting, and then we get into walking. Okay? So stages that begin at puberty and encompass the period from the end of childhood to the beginning of adulthood. This is known as, I'm sure we've all been through it. It's called puberty, a period of several years where there's rapid growth, physical changes, and accumulating in a sexual maturity. Now, the physical changes of puberty, 10 years old for girls, and it could be a range anywhere from 7 to 14 years of age, and for boys 12 years and older, and that range is 9 to 16 years of age. So puberty tends to start earlier for girls than it does for boys. Now this puberty and these pubescent changes begin with hormones surging, followed by growth spurts. Now, a girl's growth spurt they have their growth spurt between 10 and 13 of age. Girls will reach their full height at around 16 to 17, and boys will have a spurt between 12 and 15 and reach their full height around 18 to 20. Now, of course, individual variations will occur. These are milestone standards, in the same way that when you go to your doctor and you talk about milestones with your infant, you're talking about these maturational changes, and are they happening in what we would call a normal time frame. With puberty, there is also this normal time frame, but it has a range because individuality in terms of what our genetics allow us to do will occur and initiate things at different times, but that's the range. So it's during puberty that both boys and girls hit other milestones. They develop their reproductive organs and secondary characteristics and start to emerge as fully functioning and complete adults, aside from the cognitive development. So much is happening during adolescence: sexual maturity, identity, identity growth, development, a very changing time. Well, middle age is another time for changes. Two of the most important shifts in physiology for middle-aged people: one, for women, it's menopause, or the cessation of menstruation. It occurs in women usually between ages 45 and 55, but can certainly be later, and it signifies the end of a woman's capacity to reproduce. Now, men, they don't have a period, but men's experience gradually declines in their testosterone levels from about age 20, that's where their peak is at, until age 60. They may also experience a reduction in the functioning of prostrate gland, and that affects the production of semen. And so these continued changes, as most of them happen in childhood and adolescence, some of them still occur later in our life.